Uh, I'm very happy to invite Dr. Lalit Vora, and also I welcome all the people, attendees, and my fellow faculty members, my HOD, Dr. Dipti Patel ma'am, and all other students who are attending it online. I'm Dr. Karuna Nagula, Assistant Professor at Parul Institute of Pharmacy and Research. Welcome you all to the webinar Recent Advances in Transdermal Delivery. I'm extremely overwhelmed to welcome today's speaker. He is a biopharmaceutical science researcher, formulator, analyst at School of Pharmacy, Queen's University, Belfast, UK. This webinar is special for me because Dr. Lalit Vora is an alumni of current Parul University, that is Parul Institute of Pharmacy. He is 2008 pass out, and also my parent university, that is Institute of Chemical Technology, Mumbai. Uh, Also, he's student of PR Vadia sir, where like everybody knows in Parul University. Uh, he has a profound experience in pharmaceutical formulations. He has received almost 1 crore government funding for setting the scale-up facility for PLGA microspheres, so which is actually a great achievement. And he has received several honors also. The first honor which I can mention is that he had been All India second rank in GATE 2008 from Parul University. He has received British Council support for UK India PhD program, exchange program. Uh, he has continued as a postdoctoral research fellow at Queen's University. Here in his tenure, he has received Star Award 2020 and also Star Award 2021 at Queen's University for carrying out the job and carrying out extra activities outside the normal way, and also delivering the project outcomes to the industry. He has received several grants, and also he has received one of the prestigious fellowship, that is Newton Bhabha PhD fellowship. He has also been able to deliver the PLGA microsphere technology to the industry, and he is also recipient of several fundings for travel grants and also research grants from DBT, Department of Biotechnology, and DST, that's Department of Science and Technology. So with these words, I just finished this introduction, and I think we can start with the webinar. Just a minute, Lalit sir, I'll just stop sharing my slide so that you will be able to share. Yeah.
Thank you, Karuna, for kind introduction.
You can now share it.
I'm sharing my screen now. Can you see slides now?
Yes, it is visible.
Yeah. Okay. Let me... Shall I begin then, Karuna?
Yes, Dr. Lalit, you can begin. Yes.
Okay. So thank you so much for inviting me to deliver a talk on advances in transdermal drug delivery. It's my privilege and pleasure to speak my alma mater, Parul Institute of Pharmacy and Parul University now. So this is the same institute where I received All India second rank in GATE, and I was in the first B Pharm batch in this institute. So And at that time, Lalil Datta ma'am was there. She taught us during the first week microbiology. At the time, Rajesh Salla was principal. So yeah, we had a great time. Okay, I'm studying there in campus. So okay, so today I'm going to talk about advances in transdermal drug delivery. So mainly I'm working here at Queen's University Belfast on Micronel. So I'm going to talk to you about my journey with Micronel.
Sir, may I just interrupt you for a few seconds?
Yes.
Just your voice is not that clear. Just make sure that
Okay,
the voice.
let me yeah, okay. Let me change my okay. Out. So is it okay?
Yeah.
How is it now? Can you hear me clearly?
Still, there is some disturbance with your voice.
Hello.
Still, there is some disturbance with the voice.
Hello.
Hello. So there is still a slight disturbance in your voice.
Can you hear me now?
Yes, it's clear now.
Yes, I changed Bluetooth to normal earphone. So yes. So yes, so today I'm going to talk to you some of the micronal drug delivery work that I have been working on since 2017 at Queen's University Belfast. So let's start with introduction. So what is the transdermal drug delivery? Like most of you people know about the transdermal drug delivery and topical drug delivery. So what are the advantages? So this route is convenient for patient because we have the large surface of skin and body, so we can easily apply the patch or any kind of drug delivery system on the skin easily, and it's avoid the fast metabolism and therefore increase the viability and allow the controlled delivery, because skin is the barrier. So normally drug pass through slowly from that route, and that's why we get the controlled drug delivery effect, and avoid the drug degradation by the gastrointestinal tract. If we take most of medicine, we normally take by orally. So this route, we bypass the degradation from the GIT as well as fast metabolism. This is the main advantage, and also convenient for the patient. However, there are some limitations with drug delivery. We know that normally we can deliver drug, particularly like small molecular weight with the log p-value between 1 to 3, highly lipophilic drug molecules we can deliver with this drug. And another limitation is the dose. We cannot deliver more than 10 milligram per day because of the limitations of the patch size and also release rate from the skin. So main barrier is the stratum corneum. So and the stratum corneum is not allowed to pass most of the water soluble drug molecule and the large molecule. So how we can overcome this barrier? So there is a different strategies needed to overcome this stratum corneum barrier. So you can see in this figure, there is a different method. Okay, this is already explored by different researchers. So one is the injection technique by high-speed jet injection or powder injection. Microneedle. Today we are going to talk about the microneedle, different type of microneedle, and some work that already I'm doing here. There is other different technique, like we can deliver drug directly through the skin by using different lipidic nanoparticle like liposome, and also some kind of the electroporation and sonoporation techniques, and also some disruptive approaches by removing the stratum corneum completely. So today we are mainly focused on the micro patches, and this is my main focus work now.
So microarray patches bypass the stratum corneum barrier by physically breaking the stratum corneal barrier and deliver the drug directly into the epidermis or upper dermis layer, and without pain, because the needle normally we use is less than 1 mm in size, so it will not reach to the nerve system in the dermis or epidermis cell. So that way we can avoid the pain, and we can also deliver the high amount of drug. However, it's not possible directly through the patch. So there are different type of material normally people use to make a microneedle, like silicone, metal, glass, ceramic. So today we mainly focus on the polymeric microneedle, because polymeric microneedle is easy to dispose, because once we apply the polymeric microneedle, it dissolves, and then remaining part you can easily dispose and reduce the, and therefore it reduce the risk of reinsertion, because needle already dissolve in the skin, and reduce the risk of infection as well. So this is the main work we are going to talk today. So what is the microarray patches? Microarray patches are regular interspace projections, and it is highly dense, or maybe some space between the needles. So normally height of this microneedle is 150 micron to 1.5 mm in size long, and also normally base width is from 50 to 250 micron, and tips thickness is normally 1 to 25 micron, because we need the sharp tip, so it should be less than 25 micron, so it can easily penetrate into the skin when we apply this microarray patches. So there are different variables to optimize this microneedle. So number of the needles per array, whatever size of this array patch, needle height, needle base width, needle tip and shape, and spacing between the needles. So this variable we can easily change, okay, based on the requirement of the delivery system. So there are different type of microarray patches. You can see in this picture here, solid microneedle, dissolving microneedle. Nowadays, we're working mainly on nanoparticle loaded microneedle, separable microneedle, because you can easily implant the tip, particularly PLJ micro needle tips or something, and hydrogel microneedle. So hydrogel microneedle is also our expertise. Here we delivering lots of different molecules, like small molecule to protein and peptide with hydrogel microneedle. Coated microneedle, limitations is coated microneedle, we cannot coat a higher amount of drug while we load enough drug in polymeric dissolving microneedle. Hollow microneedle, in this type of microneedle, we can directly inject the solution form of the drug or any kind of nanoparticle formulation. And biomimetic microneedle. So a biomimetic is like hook. When you insert, it not easily come out. So this is different type of microneedle already well reported in literature. So today we mainly focus on the nanoparticle loaded microneedle, separable implantable microneedle, and hydrogel microneedle. So let's start with the first with the different manufacturing approach of the microneedle silicone mold and the microarray patches manufacturing. So this is the technique we use to create a microneedle mold. So normally we use the CAD file to add into the 3D printing equipment to print that particular shape or different geometry of the microarray patches. So you can see here in the right hand side different geometry of the microneedle that we already produced with this UV polymerized 3D printer. And this master template we use to create the silicone mold. Once we create the silicone mold, we can use that molds reproducibly to create the dissolving or polymeric microarray patches. So once we have that silicone mold, we can use that mold to create a dissolving microneedle. So you can see here the schematic representation of the manufacturing microarray patch manufacturing technique. Here, normally we use the dissolving polymer like PVA, PVP, or any kind of biopolymers, and we mix with the drug polymer or drug nanoparticle, and then we mix that composition properly, so we get uniform gel, and then we cast the gel on that silicone mold, and then we have to fill that mold cavities with the pressure or centrifuge. And once we fill that microarray mold cavities, and then we remove all the excess gel that is outside of this mold, and then we come up with the second layer that is drug-free gel to make only drug-loaded or drug particle loaded tip microarray patches. So you can see the light microscopy, all drug particles is only in the tips, and remaining part, remaining part of patch is without any drug. So this is the different this design that we created from the 3D printer, and this is the final microneedle resolving or polymeric microneedle look like. You can see different geometry and different height in the schematic, scanning electron microscope image. And once we have this microneedle ready, then we have to do the different characterization. So I just let you know what kind of correction technique we use at our end. So first thing, we need to see how microneedle look like. So normally we see under the normal digital microscope or scanning electron microscope. So you can see the digital or electron microscope picture here, and how whole patch look like with the different density or different height of the microneedle. So we do the different mechanical characterization as well, and then insertion in the skin as well. So in you can see it here, left hand side, this is the texture analyzer. So we normally see, we apply the different force on the microneedle, and we see the how much height reduction. So that way we can find out what is the mechanical strength look like. It is strong because we need the real strong microneedles, so we can easily insert in the skin, and also it not break while handling or during the transportation. So, and also we see the insertion in the skin by applying the different force with the texture analyzer. Also we use the paraffin as a model skin. Paraffin is the good elastic material like skin. So we use the different layer of the paraffin, and then we apply the microneedle with the different force, and we see how deep it will go into the paraffin or different layer of the paraffin, and that way we can say, okay, this is the insertion height or insertion length from the total height of the microneedle. And also we see under the optical coherent tomography, this is the live image we can see once we apply into the skin. You can see right hand side, this is the blue part is the skin, and green part is the skin, and blue is the microneedle tips, and you can see how it inserted in the skin under this optical coherent tomography. And then we also do the different release kinetic study or release permeation study with using the big full thickness skin or the thinner like dermatome skin in the French diffusion cell. So this is all the in vitro correction. Also, once we have the proper result out of this, all the correction, then we move to the in vivo study in rat or mice, depend on the drug type and the requirement of the final formulation. So here you can see the application of this microarray, different microarray patches in combination with the different nano carrier, or sometimes we don't need the nano carrier. We can directly load the drug and we can deliver through the microneedle. So application is wide, from the different skin diseases to the systemic diseases, and particularly this is useful for the potent compound like contraceptive or vaccine drug delivery, because skin is abundant of the macrophage and all the immune cells there, so we can easily give all the vaccines with this microarray patches. And also cosmetic application, and not just the drug delivery, but some uses in the diagnosis side as well. We can easily extract the fluid with the microneedle, particularly super swelling hydrogel microneedle. We can extract the interstitial fluid, and we can use for the different diagnosis purpose. So today, because Corona told me most of people joining from the research side, so I would like to explain some case study or some of the research work that we are doing here. So we are going one by one, like different work, like as I told you before. First is the nanoparticle loaded microneedle, and then implantable microneedle, and then last part we go to the hydrogel microneedle for different drug delivery. So here I'm going to tell you two different case study or research work. One is the vitamin D3 nanosuspension loaded microneedle, and other is the curcumin nanosuspension loaded microneedle. So normally we prepare the nanosuspension with two ways. One is the probe sonication, other is the media mailing. So here this vitamin D nanosuspension I prepared with the probe sonication technique, and then I loaded into the microarray patches. You can see the schematic representation of this formation work. And here you can see how this nanoparticle embedded in the microarray tips. So this is the electron microscope images. First image is the pure vitamin D3 nanocrystalline suspension, and then you can see the tips, magnified tips under the electron microscope. So all particle embedded within the polymeric matrix. So, and another important thing is, because this is a polymeric matrix, so particle remain stable for a long time. We did the stability study and we realized this is remain stable for particle size remain stable for more than 6 months. So this is the good thing with this polymeric microneedle formulation. And then other work we did with the curcumin as well, and we did the different characterization in the, also we did the skin person skin deposition study at by cutting the skin at a different thickness, and we realized a drug or curcumin nanoparticle going to the more than 2 mm deeper to into the skin. So this is just the outline of the work we did. Now, this is my current work that I'm working on, the anti-HIV drug cabotegravir nanocrystal, and also we started working with another anti-HIV drug, rilpivirine. So this is the this formulation, nanoparticle formulation, directly provided by industry. So we are not producing this nano suspension from here. This is the industrial scalable technique they have, and they provided this nano suspension. Now we are loading these nano crystals into the microarray patches. So what is the advantages of microarray patches? So this particular work is for long-acting drug, because nano crystal itself is releasing drugs slowly. So we can use this microarray patches for the long-acting drug delivery. So these microarray patches are minimally invasive and painless, can be self-administered, no need to go to clinic to inject this long-acting nanocrystals, and no sharp waste. Some introduction about HIV. So HIV is also a global epidemic, and more than 40 million people live with HIV, and most of these cases in sub-Saharan Africa. And over a period of time, HIV infects and kills the white blood cell, and that leaving the body unable to fight off certain kind of infections. So there is a new concept now, is the PrEP. PrEP means a pre-exposure prophylaxis. For that, anti-HIV medications are given to people, those are at very high risk of HIV. And this will lower the chance of HIV. And FDA already approved the oral PrEP, and it's safe and effective in preventing the HIV infection. However, daily oral pills suffer from the low adherence, and to improve the adherence, long-acting injection formulation have been developed. So this is the cabotegravir long-acting formulation already in third stage of the clinical trial. And once we inject this cabotegravir long-acting nanocrystals, it gives the 1 month up to 1 month to 3 month sustained release effect. However, some limitations with this long-acting injection. It's invasive and painful, require trained personnel, particularly in the Africa or developing world, and accidental sharp stabbing with these needles. So alternative drug delivery system is highly desirable. So then we come up with this microarray patches concept. So this is the formulation approach we use. So this is the bilayer microarray manufacturing technique, where we first cast the CAB nanocrystal loaded hydrogel, and then with the high pressure application, and then we remove the excess of this, and then we apply the drug-free hydrogel, and that way we managed to get this bilayer microneedle, that drug only loaded in the tips only. We also tried the other two types of the cabotegravir, like salt form and the free acid, for the comparison. And then we did the drug content analysis and the mechanical testing. So we found like 3 milligram drug per 0.5 centimeter square patch. So this is the high enough to deliver the enough drug intradermally or transdermally. And we found that all these three form of microneedle is mechanical strong. So height reduction is less than 6% during these different forces, like particularly 32 Newton and higher forces. Normally we need 32 Newton force to insert these microarray patches into the skin. And then we did the ex vivo characterization of this three form of microarray patches. So you can see the electron microscope images. We apply this microarray patch to skin and we observe under the optical coherent tomography. So you can see in this image, microarray, more than 50% microarray patches is like particularly drug-loaded tips is inserted into the skin. And then we run different resolution study and drug deposition study in the skin. So we apply the microarray patch in skin at different time point, and they remove the patch, and we observe under microscope to see how much drug is deposited or dissolved in the skin. So you can see the linear based within a 60 minutes, most of all the drug from the microarray tips it deposited and dissolved and deposited in the skin. And then we move to the in vivo study in rat to see the pharmacokinetic profile of this nanocrystal loaded microarray patches. So we compare with the intramuscular injection and intradermal injection with this MAP application. We collected the blood sample at different time points for 28 days. And then we analyzed these plasma samples with HPLC MSMS. So you can see here in this graph, in vivo study revealed that bilayer MAP was well tolerated and robust, and there is no any side effect during this 1 month study. All the weight of all rate are increasing, not decreasing. So this is the good thing. And it's exhibited the sustained release profile and maintained drug level above human therapeutic level after applying the single microarray patch. So this we reported first time this unique microarray design to deliver the high dose long acting cabotegravir nanocrystal. And we also try the combination approach where we loaded two nanocrystals, rilpivirine and cabotegravir, both are anti-HIV drug. And we did run the similar experimental studies, and we found the similar profile during this 1 month. Drug level is above human therapeutic level, and some of the cases, even initial 15 days is equivalent to the intramuscular injection. So therefore, this long-acting microarray patches is feasible and effective for HIV prevention, or we can say HIV PrEP. So this is the how deposition and the drug release look like from this long-acting microarray patches. So this is the schematic representation of this work. So in human, we might need to go for the multiple patches because to deliver the enough drug into the human body. And the second approach we using is the PLJ based microarray patches for long acting drug delivery. So here PLJ, most of people PLJ is the biodegradable polymer and already US FDA approved. And this PLJ is best defined by biodegradable polymer in control release of encapsulated drug and widely used for sustained drug delivery, particularly PLJ microparticle and even PLJ implants. So normally we need to inject this PLJ microparticle, or we have to surgically implant the PLJ implants. So we then come up with this non-invasive implantable drug release system with the microarray patch. So you can see this microarray patch design, and we can easily insert the tips with a microarray patch into the intradermal space of the skin. So first research case study. So this is the PLJ and nano microparticle based formulation that we developed, and we loaded into the microarray patch. This work already published in JCR in 2017. So this is the schematic representation of the formulation work. First, we prepared the PLJ and nano microparticle with the homogenization ultra-rex, and then we harvested this particle, and then we mixed with the PVP and PVA polymer, and then we loaded into the silicon mold to create this bilayer particle loaded microarray patches. So this is the particle characteristics. So you can see the particle is like from nano size to micron size. So This is the good thing to get the biphasic release profile. And drug encapsulation was more than 70%. So you can see this microscopic images where all this PLG nanoparticle particles are loaded into the microarray patch tips. And also it's possible to customize the loading of this drug loaded microparticle in the tips by changing the ratio of the particles and the polymers. Based on that, we can define the required tips height of this needles. And then we did the insertion study in skin, and we checked under the optical coherent tomography. So you can see in the paraffin layer, microarray tips are inserted well up to the third layer. So thickness of the paraffin is 127 micron. So third layer means it is more than 500 micron tips is inserted in this. And also in the skin, you can see how this drug particle or nanoparticle, microparticle deposited in the skin. And then we did the deposition study in full thickness skin. And then we sliced the skin with the microtome, cryostat microtome skin sectioning technique, and we checked the drug concentration in different layer of the skin. And you can see the drug we found up to 0.8 mm deeper in the skin. So that means this drug particle is implanted deep enough in the intradermal space. So this is the graphical representation of this work. And then second case study is M47B loaded PLJ microimplant tips to deliver this microimplant into the skin by microneedle. So we we prepared this different implantable microarray patches by using the different technique. So we prepared the first hydrogel microarray patch, and then we aligned the preformed microarray PLJ tips. So you can see in the right hand side corner how this by a 3-layer microarray patch look like. So this is the scanning electron microscope image and the optical microscope images, and also we checked the insertion in the paraffin and the skin. So here you can see how this preformed hydrogel microneedle is aligned with the PLJ microimplant tips. And this is the yellow part is the M47B. And then we did the skin deposition study. You can see here how this all the tips PLJ micro tips is implanted in the skin. And also we did the antifungal activity to see the effectiveness of this drug after loading into PLJ tips. And from this tips, drug release up to 7 days, because drug loading is really high, 75% and only 25% PLJ polymer, and that's why we managed to release all the drug within the 7 days. While the PLJ tips remain reducing the height at a different rate, you can see within the 21 days, most of the percentage, like more than 50% of the tips is dissolved. And then we did the separation study of the PLJ tips from the baking microarray patch. So you can see in right hand side how this hydrogel forming microneedle is separated from the PLJ tips in the agar media. And also that microneedle swell, so it's easy to take it out from the skin. So this is the model dry loaded tips, and you can see how it inserted in the skin under the optical coherent. So this is the work about the PLJ nanomicroparticle microneedle and PLJ tips microneedle. So then now we move to the third part, is about the hydrogel forming microneedle. So here we try to deliver the drug from the directly compressed tablet. So this is something we normally think, okay, how it possible to deliver the drug from the tablet through the skin? So this is the concept we explore here. So we use the directly compressed tablet, and then we use the hydrogel forming microneedle to deliver the drug through this tablet. So what are the advantages to deliver the drug from the skin by using this approach? So as we know that we can bypass the hepatic path, and therefore we reduce the first pass metabolism and also avoid the drug degradation, and particularly is more important with the antibiotic to avoid the antibiotic resistance. So here we use some model drug, ampersand B and levofloxacin as antibiotic, and Primaquine is also antimalarial drug, and another two drug is levodopa and carbidopa to deliver with these tablets through the skin. So this work is published in Advanced Healthcare Material. So first we prepare the hydrogel forming superswelling microneedle. So we use the Gendrez polymer and PVA as other microneedles from PVA. So this is the highly cross-link microneedle, so it is normally swell and it not dissolve during the application time. So you can see in the OCT images how this needles inserted in the skin. And we did the different correction technique, like swelling study of this hydrogel microneedle and mechanical strength and insertion study of this microarray patches. And then separately, we formulated the different tablets from this different drug, like ampersand B, levofloxacin, and Primaquine, as well as levodopa and carbidopa. You can see the formulation, like we use the normal tablet excipient like MCC, mannitol, starch, crosspovidone, and all this different excipient. And then we did the mechanical strength of this tablet. So we prepared this tablet with normal IR tablet punch. So this is how this tablet look like. And then once we produce the tablet, then we combined with the preform hydrogel forming microarray patch. So you can see how this microarray patch with the tablet look like. And then we did the in vitro permission study with this different formulation. You can see the release kinetic from this different tablet. So first is the ampersand B release from this tablet hydrogel patch. So within a 24-hour, more than 15 milligram drug is delivered from this 180 milligram loaded drug load ampersand B loaded patch, and similar we found a good amount of deliver with other drug, levodopa and carbidopa. We managed to deliver 40% out of this tablet, and levofloxacin around 30%. And then we went for in vivo study where we applied this tablet with microarray patch to the back of the rat skin, and we collect the blood sample at different time for 48 hours, and we check the plasma level of these different drug molecules. This is the schematic represent how we do the animal study with this microarray patches. So this hydrogel microarray patches remain as it is even after 24 hour application. It just swell and then it not remain in the skin. So this is the good thing. So there is no any toxic residual of this microarray in the skin. So here you can see how this different microarray patches in compared to the oral drug delivery look like. So we managed to deliver more amount of drug from this 4 patches, particularly in levodopa, carbidopa. And also you can see in amoxicillin compared to the oral Gavals, AUC is the 2 times higher than the oral Gavals. Even the dose is similar. And similar trend we found with the levofloxacin in the rat study. So conclusion of this work. So MEF has been explored by many different researchers for different applications from drug to vaccine. So here we mainly working with the high dose drug, hydrous drug, and also for the hydrophobic drug like nanocrystal or even the PLG based implantable drugs. So dissolving microarray system. So as you have seen in the different slides here, to deliver the nanoparticle transdermally in vitro and in vivo, and it capable to deliver even the high doses of the drug through this different microarray pages. So further to this, like we require the collaborative effort between the academia and industry to address the concern of the particular safety concern of this microarray page and manufacturing, because still it's not easy to produce this microarray pages at large scale, and then also acceptability of this microarray pages for the general public. So thank you so much for the listening. I am happy to discuss and happy to answer the questions from the audience.
I have a question that
Yes,
last study, that is the microarray that is attached to the tablet.
Yes.
Right. So how the tablet that is attached to the microarray?
Yes. So it is we applying this tablet. Let's say we first apply the microarray patch to the skin, and then we put the tablet top of that, and then we come up with the adhesive layer, so tablet not move during the application. So this is something, it is not like already attached. We have to attach when, or we have to apply the tablet top of the microarray patch when we do the any kind of experiment or skin application, or even during the animal study. So, but it is also feasible to apply the patch by some kind of polymeric adhesive layer. Okay, that is soluble. It is water soluble when that's micronal swell and drug start release from the patch.
But that is possible into the research, right? But whenever we want to transfer it into the market, at that time we cannot say to the patient that you have to apply first that patch, microarray patch, and then you have to put that tablet on that. So
yes, you are right, because see, this is the proof of concept, right? So at initial research level, we have to come up with some sort of design where we can easily run different experiments. So if you see in this in vivo setup, okay, we use the microform tape with the aluminum foil, okay, to hold the tablet on this microarray patch. If you see the this schematic representation there, so this kind of patch is possible. Like this is my microform with adhesive layer, so we can attach, okay, in this small tablet size hole, we can put the tablet in between there, and then we attach the back side of the microarray patch, and then that remain as it is, like so this kind of strategy might work.
Okay, and one more thing, that is we all know that the drug which is present into the tablet that start releasing when it comes with contact with the fluid.
Yes, so yeah, you can see in the release profile, okay, here there is a lag time, because most of cases drug is not releasing immediately, because the microarrays, the super swelling hydrogel micronal normally take off interstitial fluid, and then the fluid pass to the tablet, and then tablet start releasing drug, and then it's go to the skin. So after 2-3 hours, there is enough interstitial fluid come up to release the drug from the tablet, and then it start moving from this microarray patch to the skin. So that's why drug release is slower initial phase, and then it pick up to the higher level.
Okay, and one more thing, that is the paraffin we have used so how that we have to perform.
Yes, so this paraffin, the normal laboratory paraffin, there is a thin layer. It hardly thickness is 100 micron, 125 micron. So you have to cut the small pieces, and then you apply all the paraffin one by one top of each other, and then let's say 10 or 15, okay, that layer, and then you can use that combined layer to apply the micron or to check the insertion depth, and then you can peel the layer one by one, and you can see the hole, how deep it's go to different layer. Let's say first layer, all the 100% needle is penetrated. Second layer, maybe 80%. Third layer, maybe you can see hardly 50% hole or less than that. So you can count that hole, and based on that, you can say my needle is penetrated to that layer, and the thickness of this layer is this much, so how much is inserted and how deep is inserted. So you can easily calculate that things.
Okay, thank you.
Yes, I do have one doubt.
Thank you. Yes, Karuna.
What is the like? There can be some immunogenic responses also, like on this the people who are having sensitive skins. So, like when you apply these patches, like for sensitivity testing, they apply some patches, doctors, for checking the allergies in the people. So, like, will these patches be safe? And this is one doubt. And the second doubt is, can we develop a patch for COVID like this, rather than taking every year the shots of vaccine? Can we have a
Yes. So
patch?
if you,
Just a crazy
yeah.
question,
So
I know, but yeah.
thank you for asking this question. Really interesting question. So, second question, yes, if you, we also working with the mRNA vaccine at our end, and we're running some of the animal study. Okay, so yes, because vaccine is like potent. Potent means hardly we need some microgram to load into the microarray page. Okay, even we loaded 3 milligram drug in the 0.5 centimeter square. So vaccine is the small, like definitely molecularly large, but dose is small. So it's possible to load enough vaccine dose in small size of the page. And also, you know that we can easily get more immune response through the skin instead of injecting intramuscularly or subcutaneous, because more immune cell available in the skin or interstitial in epidermis layer, right? So yes, there are some group also working directly with the COVID vaccine as well, and they produced some of the nice work recently, and some of also in clinical trials, some study in clinical trial as well. So yes, it is possible to give the vaccine with microarray patch. So the advantage with microarray patch, let's say first booster dose, you might need to go to the clinic and by injection, but remaining booster or other doses you can easily take with microneedle. Okay, and also particularly this is important, microarray patch, you can stabilize the vaccine, no need for the cold storage. So you can just, you can solve the supply chain problem because your vaccine is embedded in the polymeric matrix. So that's, and also if you use some of the polymer, like some biopolymer, you can easily stabilize the vaccine. So that way you can even just post your vaccine to the some villages where there no any refrigerator or any things available there, right? So and also patient can, or not patient, because we're giving vaccine to the normal people can easily apply at home. Okay, because you just need to press enough, so insert all the tips in the skin. So yeah, this is the interesting thing, and people now looking into this as well. And first question, say microarray patch is safe. Yes, it is safe because we proven at our end. We did some human clinical studies. Okay, we apply the patch to the different people, and we see the any immunogenic reaction. Like we collect the blood and we check any biomarker or like any immunogenic reaction or not. So till now we found this all the microarray patch that we're using is safe. In case of the hydrogel microneedle, because we are not depositing any polymer, okay, we just that microneedle swell and these tips remain intact. So we are not depositing any polymer, so that way is also safe. Dissolving microneedle, we have to use the biocompatible polymer. So if we depositing any polymer there, okay, it is even if go to systemic circulation, it is okay easily. If we use low molecular weight, then easily extract from the kidney. And regarding checking the like as you said, okay, yes, we can use the microarray page to see the any immune reaction, like allergic reaction or immune reaction with the different. So people use the loaded with different allergen, like peanut. Some people have peanut allergy, so they loaded peanut antigen, like peanut particle, and they check. Okay, so because you can easily insert, okay, or implant any kind of allergen to the skin, and you can see the redness there. So you can say, okay, this person is like this. So this way, yeah, microneedle is a useful tool to check the any kind of allergy as well.
Thank you so much. Is there anyone else who will, like, any doubts, any queries? Okay. Thank you
I
very
think
much.
we have more 10 minutes, so in case anyone need to
10
discuss
minutes.
anything further.
Yes, yes, yes.
Lalit, Lata ma'am is there?
I think ma'am has not joined.
No, Lata ma'am is not there.
Okay.
Okay, so Lalit, you have to come to Parul University whenever
Yes,
you are
whenever
in
I visit,
Baroda.
next time whenever I visit
Yes,
India,
you have to
definitely
have to
I
visit.
would like to visit the campus. I last time visited in
Yes.
2000.
I think there's some issue with your voice. Hello? 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 Hello?
Oh, so I think
Yeah, the voice was not audible. Your voice was.
I don't know why my okay, speaker is going to mute.
Okay. So
Okay, that's fine. Okay,
thank you. It was actually an interesting topic. Such a good research work you have done, and that is so fine and precise. Uh, so, like, we are very thankful for accepting our invitation, and we would really like to see you in person
same here.
whenever we have any further
Yes,
events to share your research. Now, I would just request you to stop sharing the slide so that I can share the certificate
yeah.
as a token of gratitude from us. Dipti ma'am, can you just, I'm sharing the certificate.
Yeah, yeah,
Just
yes, yes.
think that you have given it.
Okay.
Kindly accept this e-certificate as a token of our sincere gratitude. Thank you.
Oh, thank you so much, Dipti ma'am. I really enjoyed talking with all of you, and looking forward to
Yeah,
talk to you all soon.
yes. Thank you.
Thank you. Yeah. Bye.
Thank you so much. Thank you
Thank you,
everyone
Karuna. Thank
for.
you.
Yes, you can. Yes, Lalit, if you want to share something, you can share.
No, no, I'm just thanking you because thank you for organizing this and inviting me to talk to all of you. Okay. And nice to see you. Okay. After a long time as well.
Yes.
Okay, so with this, we conclude the webinar. I hope you all have got really good insights from this. And so we would like to conclude this session here. Thank you, everyone.
Thank you.
Yeah.
Bye.